Hello, listeners. Yamina here. Welcome to this episode of the Dr. GPCR podcast. I recorded this episode with Dr. Bryan Roth from my car. It was parked in front of a Wi-Fi spot near my home last July. During this episode, you'll hear Bryan refer to my improvised studio. Bryan is the Michael Hooker Distinguished Professor of Pharmacology at the University of North Carolina, Chapel Hill School of Medicine. Originally from Montana, Bryan has an MD and a biochemistry PhD degree he earned at the St. Louis University in 1983. He also trained in pharmacology at NIH and in molecular biology and psychiatry at Stanford. Witnessing the psychotic break of a relative at a very young age influenced Bryan's path. While still in Montana, a lecture from a visiting professor on neurotransmission ultimately sealed his interest in receptors involved in psychiatric diseases. Before we jump into this episode, I wanted to let you know that our October newsletter is now available. Please visit drgpcr.com/newsletter to subscribe and share our newsletter with your colleagues. This month's newsletter was made possible by Twist Biopharma. If you'd like to support and sponsor us, please visit drgpcr.com/sponsors or email us at hello@drgpcr.com. And now, let's dive into our episode with Dr. Bryan Roth. Hello, everyone. Welcome to the Dr. GPCR podcast. Today, I have the pleasure of having with me Dr. Bryan Roth. He is a distinguished professor at the Department of Pharmacology at UNC Chapel Hill. Hi, Bryan. Welcome.
Hi, how are you? Greetings.
Good. I'm happy to have you here today.
Yeah, it's great. Looking forward to this.
Absolutely. So I briefly introduced you. I think the GPCR community knows very well who you are. Would you be able to tell us a little bit more about your trajectory as a scientist that led you to where you are today?
Yeah. So it's actually started when I was quite young, and I've mentioned this before in interviews. So a member of my family had schizophrenia, and their first psychotic break was when I was 5 or 6, and this had a huge impact on me at the time. And I would say from the time I was probably in 8th or 9th grade, I decided that what I wanted to do was be a psychiatrist and come up with new ways to treat mental illness. And the other thing around that time was it was sort of the tail end of the 60s. So I grew up in Montana, so we were buffered from a lot of the things that were going on in the 1960s, but not entirely. And I also got interested in you could see basically people that would take psychoactive drugs. They would have this huge effect on them. Yep. And so these two interests basically converged when I was in undergrad in Montana. There was so I went to this really small college in Montana. There were, I think, a total of 1,100 people.
Wow.
1,100 students in all 4 years. So very small, and no research going on. But it was good for pre-med. And at the time it was, so I was clear, I wanted to study the brain. I wanted to study drug action on the brain, but there was no way of doing that coming from a small town in Montana with no scientists. And so I decided, well, I'll go to medical school. The last year or so, we had a visiting scientist from the University of Oregon who came in. I think he was actually coming, it was in the spring, I think he was probably coming to Montana to go fly fishing.
Oh.
And because we have this world famous, trout streams basically all over the northern part of northwestern part of Montana where I was in college. And as part of his sojourn, he gave a lecture on neurotransmission. and he discussed the concept of receptors.
Wow.
And so that was around the time that radioligand binding technology had been invented by Saul Snyder and Candace Pert. And I said, that's what I want to study is receptors. That's it. I'm going to study receptors. I don't know what they are. They sound really cool. So, so that was the start. And then I got into medical school. What I tell people is after a day or so, I realized I had made the biggest mistake of my life. And I wasn't really sure if I wanted to practice medicine. And so I started looking around for other things to do. And it turned out there was somebody in the biochemistry department who was studying receptors. And he was basically the only person in the entire medical school studying receptors, by a guy by the name of Carmine Kasha. He's now retired, but fairly well known in the opiate receptor field. And I said, I want to study receptors. Can I work in your lab? And so he let me work, and I, spent basically all the time I should have been going to classes working in his lab. And then finagled my way into the MD PhD program, and ultimately got a PhD on opiate receptors, and then I was hooked. So, and at the time, it's interesting to think about this. So this was in the early 80s, late 70s, early 80s. There was debate in that era, believe it or not, whether receptors were proteins.
Wow.
And in particular, whether the opiate receptor was a protein, because nobody had ever purified it. And there was actually a group of investigators who said the opiate receptor was actually cerebroside sulfate, that it was a lipid.
Well, okay.
Okay.
That was my next question. So if they thought it wasn't a protein, what was it?
Right. So there was this debate in the field. Is it a protein? How many opiate receptors are there? There was pharmacologic evidence for at least 2 or 3, and so it was a very wild time. And the idea of G protein coupled receptors was not a concept at the time. Basically, it wasn't until rhodopsin was cloned, and then beta receptors were cloned basically by homology with rhodopsin, that it was realized by the folks in the Lefkowitz lab that this was a huge family of G protein coupled receptors. And then, ultimately opiate receptors and other receptors were cloned. But it was sort of a wild time back then. Nobody really knew. We didn't really know what was going on. I was in the biochemistry department, and I think half of the faculty thought what I was studying was an artifact because I couldn't purify it. But I published, a nice paper in JBC, which at the time was a pretty good journal. Still is a great journal, but at the time was like a really, really good journal. And so they gave me my PhD, and that was it. I was hooked. I've worked on them ever since.
So that makes my question, what is your favorite GPCR, obsolete? Clearly it's opioid receptors.
Actually not. I started out studying opiate receptors, and I won't mention any names of people living or dead, but at the time that field was populated by a cast of very colorful characters. I'll just put it that way. Mm-hmm. And there was a lot of, you would go to these meetings, and there was a lot of people yelling at each other.
Wow. Because there was this competition between them?
Well, they didn't agree. Was this subtype of receptor a real subtype? And there was this idea actually that one subtype of opiate receptor was coupled to guanine nucleotides, and another subtype was not.
Okay.
Okay.
This was all before these receptors were cloned.
all before they were cloned, yes. And lots of other crazy stuff like that. And so I decided after I got my PhD, I never wanted to study opiate receptors ever again in my life. That changed, some years later. But it was basically because, these people, they seemed like they were crazy to me in some ways. They were clearly very passionate about their research, but it was just, there wasn't a lot of data. It was just, I think, this class is not coupled to GTP and this class is basically. And people would argue about whether the data was real or not. So, when I did my postdoc at the NIH, I started studying serotonin receptors. And it turned out as luck would have it, the receptor that I started studying turned out to be the receptor for psychedelic drugs, which, I was very interested in their actions. And also the receptor for many therapeutic medications, so atypical antipsychotic drugs, and some antidepressants and so on. So just by dumb luck, I happened to start studying a receptor that ultimately has turned out to be, interesting and important, but through no particular effort of mine, just dumb luck, I would say. So
that's great. That's great. Getting back to the era of dopamine receptors and all these inconsistencies in the field. So you had mentioned that at that time, radioligand binding was the brand new hot technique to measure receptor function. What were the other techniques, or were there any other techniques that people were using to at least measure the effects of receptor activation?
Yeah. So when I was a graduate student, there really wasn't anything else other than binding assays, basically. Gilman had, Al Gilman had worked out this cyclic AMP binding protein assay to measure adenylate cyclase activity. And after I'd finished my PhD, I did it wasn't a formal postdoc, but sort of a semi-formal postdoc while I should have been doing other things related to medicine. And I discovered the secretin receptor. And we did that by radioligand binding. And then there was a scientist there, Lynn Hallett, who had come out of Al Gilman's lab, who had brought the adenylate cyclase assay technology with her. And we showed it was GS coupled, basically. So I tell all the people that study family B receptors that they should remember that I'm the person that actually discovered the first family B receptor. I published one paper on it, and from a neuroblastoma cell line, and showed it was GS coupled. Yep. But that actually, the cyclase technology in those days, it was, for reasons that are not entirely clear, it was difficult to do. It was, required this double column thing.
I've done some, interestingly, when you had to, starve the cells, load them with tritiated tracer, they would make the tritiated cyclic AMP that would then lyse the cells and run the cyclic AMP were produced through two sets of columns. So we had one set, you ran your lysate, you washed, and then you put the second set on the second column, and then you collected something that was right before the EPAC BRET biosensor came out of Michel Bouvier's lab. And I think before, in between those two, we had this this kit that didn't necessitate using all these columns. You could you had this charcoal method. But I do remember that. It was quite a pleasant experience having to fill those columns.
Yeah. Luckily, Ellen did that for us, so I didn't have to do it. And then when I was a postdoc, I basically discovered, or was part of the discovery of metabotropic glutamate receptors. So I sort of, I was in Herminio Costa's lab. And at the time, there were 50 postdocs in the lab, so it was this gigantic lab. And my project was to develop phosphoinositide hydrolysis as an assay system, which was just, it was basically a pathway that was just being discovered. And so I did, and I showed that some serotonin receptors were coupled to it. And then we discovered basically metabotropic glutamate receptors, other receptors. So then we had basically two sets of assays. There was for functional activity, other than organs and organ baths, right?
Wow.
So I actually did some of that research as well. Yeah. But there was just radioligand binding, and then at least you could measure what we now know are GQ coupled receptor activity, and then the GI and the GS. But even then, so this was the 80s. It was for reasons that are still I would say still mystifying to me. So I was trained as a biochemist, so it was fairly straightforward for me to develop these assays. But for pharmacologists, it was magical, I think, to some extent. It was just a different era, basically,
Yes, very far away from the, plate-based 384 well plate assays where.
Yeah, 384 or
There you
1536.
go.
Yeah.
You get so much data out of one plate that you can play around for a week unless you have some bioinformatic magic.
Right. Yeah. Things have changed a lot. Yeah.
So it's the 80s. You're at NIH. You're working on serotonin receptors.
Right.
What next? You're an MD at this point. You completed
MD PhD.
Okay.
Yeah. Since I was poor in medical school, I got quote unquote a scholarship from the Navy to pay for my MD training. My PhD training was I got a graduate fellowship. And so I went to DC, and I then had to pay the Navy back. So I did a year of a psychiatry internship at Bethesda, what was then Bethesda Naval Hospital. And, when I should have been sleeping, I was working in the lab at the NIH, basically. And then the Navy let me do 4 years of research as payback. Yeah. So I continued doing some work on serotonin receptors. I ultimately had my own lab in the Navy, basically across the street from the NIH. And then also did work on, since it was the military, they wanted some military applications. So I studied septic shock, which was a frequent cause of injury and death in military personnel, and actually studied receptor desensitization and functional selectivity.
Wow.
So I wrote a review article in '87 laid out the case for GPCR functional selectivity using serotonin receptors as a model system. And the idea that I sold the Navy was there are these multiple pathways. We didn't know about multiple G proteins at that time, but it was clear that receptors could couple to multiple signaling events. And I said, we can make a drug that activates this pathway and not this pathway. And maybe this pathway will not be desensitized. And so we can come up with a drug that's better for sepsis than what we have. And so they let me work on that, and I learned some molecular biology along the way, and so that was very helpful. I published a book on septic shock, and sort of became one of the world's experts in that area. And then I left the field when I was done with my time in the Navy.
Back to GPCRs and
right.
And working on that. And then after that, you got your position at UNC at the Department of Pharmacology?
No. So then I went to Stanford and finished my psychiatry training. It was clear to me at the time that I was not going to make it as a scientist. There was no way I was going to get a job as a scientist, basically. That's at that time. Okay. That was clear. Okay. My publication record was just not particularly strong, and I actually wasn't ready to run my own lab either. I needed to grow up. So I decided, well, since I'm not going to make it as a scientist, since, I may make it as a scientist, but it's not clear that I'm going to be able to make it as a scientist, I better have a fallback here. And I was always interested in psychiatry. So I went to Stanford to do my psychiatry training. And I would say, as is typical of me, when I should have been sleeping, I was working in the lab, learning molecular biology.
That was my next question. So at Stanford, did you ever get to work in a lab? But I guess yes.
Yeah, I did. I wasn't very successful, but I was, it's funny. So I tell this story to people. So I was so unsuccessful as a scientist at the time that at the time when I was in training, I got a fellowship because the guy that was mentoring me was head of the selection committee, and it was a very prestigious fellowship, the Dana Foundation Fellowship, and at Stanford University, so it was a big deal. And every year we would bring in a famous scientist to give a talk, and then he would meet with all the Dana Fellows. There were 5 or 6 of us there, and give us advice on how to be successful in our careers. I won't mention the name of the person, but he's a really famous scientist. Now, I like to joke, I think I have more citations than he does, though. But at any rate, when he when he met with me, he said, "Bryan, I'm going to tell it basically, I'm going to give it to you straight. You're never going to make it as a basic scientist." He said.
Okay.
He said, "You've never published anything important." And he said, "Based on what you're doing, you're not going to publish anything important, and you should just give up now. Basically, give it up entirely. You're never going to make it." He did say it's possible that I could run clinical trials for a pharmaceutical company, though, because I knew pharmacology quite well, and I could make a comfortable living and be involved in some interesting stuff. But the chances, I should just forget about basic science altogether. It was just not in the cards for you at all.
Goodness, I've heard stories like this, and yeah, I think yeah. Well, that's. I'm glad you didn't listen to that advice.
I was too stupid to listen to him, basically, is what I tell people. So, so I did. So by then I had my, my clinical training was done, and I went to Case Western. So Case Western Reserve University, the psychiatry department hired me, and they gave me a lab which was about as big as your car. Okay.
Well,
maybe slightly bigger, but about that size. And then my job was to basically run a clinical ward. So I, was interested in schizophrenia, and I ran a schizophrenia research and clinical ward for many years until my lab finally, got off the ground, basically. Okay. When I was starting out at Case Western, it was very, very, I tell people it was very, very difficult for me to get started, extremely difficult, because I didn't have any publication record of note. And I couldn't write grants that were interesting to anyone. So the first grant that I submitted, so this was in the era before they had triaging of grants. So now, 50% of the grants are triage. So my first grant would have been triage, but they gave me a score because they didn't have triaging then. But basically, so I subsequently have found out who it was that reviewed it, but I won't say their name. But basically said, the same thing that I was told at Stanford. This person has never discovered anything of note. And based on what's in this grant, we'll never discover anything of note in the future.
Oh God.
So pretty rough stuff. And so I called the program officer and at the NIH who was handling the grant, and he said, you know, Bryan, maybe you should consider a different career other than science.
Wow. And still you didn't listen.
I did not listen, no, because I just really loved
Yep.
working in the lab and what I was doing. And you know, all these receptors were being cloned, and it was a very exciting time in research.
This is in the 90s, early 90s.
90s, yeah, early 90s. So lots of, you know, all the
Yep.
receptors were being cloned, and you know, it was really fun, a fun era. And you know, I submitted a revision, and it actually got a better score. It wasn't good enough to get funded. And my program officer said, don't ever send that grant back to us again. They don't want to see it again.
Oh goodness. Was it about GPCRs?
Yeah, it was structure and function of serotonin receptors.
Wow.
Yeah.
I think you were ahead of your time, probably.
Yeah. So molecular modeling, mutagenesis, I proposed, you know, will purify the receptor, try to crystallize it, this sort of thing. And my chairman actually said to me that I wasn't going to make it, basically, you know. So I sent it in a third time. It got a 5.4 percentile. So a very good score. Okay.
Right.
That's a good score, right?
Probably.
It's a very good score. Okay.
That's awesome.
It was an awesome score. They felt sorry for me.
They just didn't want you to submit it a fourth time.
They didn't want me to submit it a fourth time. They didn't want to have to see it again. And they didn't fund it.
Wow.
Because they said that year they were only going to fund the top 5%.
Oh.
So I was at 5.4. So I knew somebody in another institute and told him my tale of woe, and he said, well, I can't handle this, but he said, I know somebody in the institute that, you know, studies receptors, and
yeah.
You know, they funded up to like 20%. So they were happy to take the grant, and then I got funded, and then, you know, I've been funded ever since. So yeah. So it was very tough, very, very tough for me, really hard. Yeah.
But you persevered, and that's because you were, at least from what I can hear, very passionate about the science.
Yeah, I love doing science. Yeah. And I didn't listen to what anybody said.
Yes, sometimes that's also very important. You can listen to what people have to say, but you still have to do what feels right, and especially when it comes to something you're passionate about.
Yeah, and I got lucky, you know.
There is also all the time that component, but I think being able to not give up at the first try is also very important.
Yeah.
So how did you end up at UNC?
So you know, when I was at Case, you know, things started going well, and I ultimately moved to the biochemistry department, became a full professor there. And then my daughter was ready to go off to college, and my wife said that she didn't want to live in Cleveland anymore because she was tired of there not being any sun. So in Cleveland, there is no sun, basically. It like 90% of the time is cloudy.
A little bit like Montreal sometimes.
Yeah. And so she basically said something to the effect that, you know, she said she's going to go someplace sunny and I can follow her. So I was actually happy at Case Western. Things were, you know, great, and people were very nice to me, and I had lots of friends, and, you know, the research was going well. And then Gary Johnson from UN. So I let people know that I was looking to go someplace that was sunny. And Gary Johnson, who was chair at UNC, basically just called me up on the phone, and he said, I'm going to give you an offer you can't refuse. That's what he said.
Wow.
And he told me what it was, and I said, "You're right, I can't refuse that offer." So they gave me a really good offer. It's sunny 90% of the time down here, and very nice. So
Can you share some of the details of that offer?
no, I can't. No, no, I can't.
That's okay. That's
Yeah.
okay. Well, at least you got.
I got an endowed chair. That was
Wow,
nice. Yeah, that was really nice. Yeah, yeah.
that's great. And you got the sunshine.
And I got the sunshine. Yeah, and we moved into a brand new building,
Mhm.
research building, which is beautiful, just spectacular. And I've had lots of fun here. Lots of great students, great postdocs, great colleagues. So it's been lots of fun.
That's fantastic. And since so for the past decade, what would you consider as your favorite receptor or family of receptors?
So it's usually whatever one we're working on at present. Um, so I actually got back into the opiate receptor field, as you know,
Yep.
and we solved structures of opiate receptors and all this stuff. And that was, I would say, getting the first structure, that was huge. Getting our first structure, that was really big, because it was what I was always, as I told you, I was interested in drug action at receptors,
Yeah.
and really wanted to see what this looks like at the atomic level. And so when we started getting those structures, that was just amazing, and it continues to be amazing. I mean, we have so many structures now that we're writing up. It's just incredible.
Great. And it was a
And
special month last month. I put together this newsletter on a monthly basis, and I think we counted 8 or 10 papers that just came out just in June
yeah,
that had to do with structural cryo-EM or structures,
yeah, yeah,
various GPCRs.
yeah, yeah,
And the field
yeah.
has been booming structurally speaking.
Yeah, it's amazing. And then structure-based drug discovery and all this stuff is just, it's been awesome to really be part of that. We're doing a lot of work on serotonin a lot of work on. So I have a huge lab right now. There are about 30 people in the lab,
I saw.
and probably 15 are working on serotonin receptors. So we have a huge effort on serotonin and dopamine receptors, and then all the other receptors basically. So we study them all, but right now we're really interested in serotonin receptors in particular, and dopamine receptors. So yeah.
More the structure function relationship of these receptors, or is there any clinical context in which you're
Yeah. So
interested?
the idea I've always had, uh, I would say, which is not a novel idea, was that if we had the structure, that would be helpful for developing better medications. Um, and so the idea always is that once we get the structure, then we use it for drug discovery purposes. So most of the structure studies there's a component to solving the molecular determinants of bias signaling, or functional selectivity, and then using that information to inform drug design and ultimately therapeutic drug discovery. So we have lots and lots of projects with those sorts of aims. We do some animal studies. So I have a big lab. There are probably 3 postdocs that are working with mice. So I have a big mouse bill. And we just a paper just came out in Nature Neuroscience earlier this week. So I invented RED technology, and that of course is really taken off. So we have a new ligand that works in mice and in monkeys, and works as a PET ligand, and will be very useful for translating that technology going forward. So just lots of things. We have so many projects ongoing now, and I have so many collaborations that I've had to tell people in the lab to when we have lab meeting to remind everybody once again what their project is.
30 people, yes.
Yeah.
Definitely. Send out notes the night before so that everybody can get a chance
Right,
to read
yes.
what the project is.
Now, what's this project you're working on again?
Yeah. So in the context of the serotonin receptors, you're looking at structure-function relationships, understanding structurally how the receptor works, and you want to develop better drugs.
Right.
What are you looking for exactly in this context?
So,
what are the benefits of the current drugs?
Yeah, so one of the most, I would say, one of the potentially most amazing projects we're working on is I just got this big grant or award. It's called a cooperative agreement, technically, from DARPA, which is the Defense Advanced Research Projects Agency, which they're notorious for coming up with impossible ideas that will be revolutionary. So they came up with this idea, I think in part after seeing one of my talks. So there, a little bit of background here. So certain psychedelic drugs like psilocybin and LSD are showing really amazing efficacy for treating depression. So psilocybin actually in the US is in phase 3 clinical trials and has been given breakthrough status by the FDA for treatment of depression. And basically, people take a single dose. They have this profound psychedelic experience, and then their depression is gone, basically.
Wow.
For weeks or months. So they follow these people for up to 6 months now, and most of them are depression-free.
Interesting. So
one dose. Okay. Now, that's great, but you're taking a psychedelic, right? Not everybody can take a psychedelic, and those are not without their problems. So the idea is, can we make a drug that is not psychedelic, that has the same therapeutic effectiveness?
Yeah.
Okay. So that's the idea. Make a non-psychedelic psychedelic.
Got it.
Okay.
A biased non-psychedelic.
Basically biased, a drug that's biased for particular signaling pathways that are therapeutic and not psychedelic. Okay. Can we do that? Well, we can make biased drugs, yes. That we can do. Will they be therapeutic? We don't know. So
exactly.
So it involves, solving structures, doing structure-guided drug discovery, optimization, a huge amount of chemistry, and animal studies, basically. So it just started. We just did our 6 week report, so we're 6 weeks into it. So, but it's moving along. It's moving along amazingly well. So.
That's great. And do we know if the psychedelic activates differential signaling pathways at the serotonin receptor?
I can't give anything away, but I don't know. Have you ever watched this show Billions?
No, it's funny because my husband was watching it just last night.
Okay, so one of the main characters in Billions has this phrase. What is the phrase, Judith? I am not.
I am a master.
No, I am not uncertain.
I am not uncertain.
Right. So the venture capitalist or investor has this phrase that he uses when he has inside information on something that no one else has.
Okay.
And he'll say, I am not uncertain.
Okay.
Okay.
Got it. So you're not uncertain.
I am not uncertain. Let me just put it that way. There may be data out there that's unpublished that we may have in our possession. So
got it.
That we are the only people in the world that have in our possession. So yeah, I think so certainly we can make drugs that are biased for any pathway. Okay. if you have sufficient medicinal chemistry resources and appropriate assays, you can make a drug that will do anything, basically. The question, though, is the pathway the one you want? Okay, right? That's the question. And I'll just say I'm not uncertain about that.
Definitely. I mean, it's a big question in the field. For example, with the opioid receptors, when for quite some time people thought, well, you need to activate GI but not beta-arrestin, and then you'd get the analgesic effect but not the side effects.
Right.
Now it turns out that it's not as clear cut.
Yeah, it's not. And I think a big part of the problem is we don't have very good tool compounds. So the compounds, so all the drugs that people say are quote unquote biased.
Yeah.
In our hands, they're not particularly biased, basically.
Uh-huh.
And we actually have published this, sadly, in the supplement of our paper. But it's published in Nature. We show that they, if you do the assays with GRK and all the things that you need, then none of the drugs are particularly biased to any great extent. And that's the case actually with a lot of the so-called biased ligand programs, or functionally selective ligand programs. The compounds never were particularly biased, and so really, and there are lots and lots of these programs out there. It's not just virtually every receptor has a story related to that. But I think we've been hampered to some extent because a lot of the assays aren't particularly good. We've actually moved entirely to BRET technology.
Okay.
I've become a real fan of BRET technology.
I'm a fan too, so
yeah.
I grew up with it.
Yeah. So it was one of these technologies that it just seemed difficult to do. I don't know why that is. I think part of it was the readers weren't very good.
Yes.
Right?
Agreed.
You would agree with that? Yeah.
Readers weren't very good, and depending on some readers, sometimes you had trouble measuring the fluorescence if the cells had autofluorescence, and then it just messed up the signal. But I think it's a beautiful technique because you're looking at a ratio.
Yeah, it's ratiometric. It's great.
Yeah. I love it.
Yeah.
I love it.
Yeah. So the readers now are really good. So I won't, I'm not going to mention any brand names, but we just got a reader that we do a 384 well played in 45 seconds.
Wow. That's fantastic. Yeah, compared to old school readers when you needed 3 minutes to read one time the 96 well plate.
Yes. Yeah. And usually, so with the old reader, we usually would have to measure the plate 5 times.
Yes.
Right? Yeah.
Exactly.
Because it just wasn't very stable.
Exactly.
So now you can get a very stable read in less than a minute for a 384 well plate. Yeah. And we're in the process of buying another amazing piece of equipment that will read a 384 or a 1536 well plate in 2 seconds.
Wow. Wow. And then you need a whole bioinformatics team to process the data as it comes off the.
Yeah. So I have dedicated people to analyzing data, but so. And we've optimized, we put a lot of work into optimizing sensors so that they have better dynamic range and stability in that. And it's basically, I consider it definitive assays now,
And they're easy in the sense that once you have the best dynamic range possible, you have the ratios figured out, then you just have to put it in your cells and then throw any possible compound you can think of at it, read it, and then process it kind of like in a factory where you can gather a lot of data in live cells.
Yeah. In real time.
And yeah, exactly. exactly, which I think is really wonderful.
And there's not a lot of downstream amplification
No,
either. So they're great assays. You do need a specialized reader though. That's the problem. Yeah.
you do. I consider it, well, you do need a specialized reader, but I do consider it, once you get the hang of it, to me, it's an art
Yeah.
of doing these experiments in a way that, you can, as a lab person, that it takes 2 minutes to do this, and then it takes 5 seconds to plop it in, and then you can plan out how many compounds you can run in the next hour, because
Right.
everything is like clockwork.
Yeah. So
And then you can
I tip my hat to Michel Bouvier every time I see him. I say, because I remember I went the first GPCR, the Canadian GPCR meeting, or Great Lakes GPCR meeting. I think he unveiled that probably in 2004. It was before he published anything on it.
Yes.
And at the time, he said, BRET, bioluminescence resonance energy transfer. And I said, wow, this sounds really cool. And we went back to the lab and tried it, and we didn't have a reader. We had basically a spectrofluorimeter, and we were trying to, adapt it. And I said, this is just not going to work. How do you get this?
Definitely not the best for that.
The machines have really caught up, and it's an amazing technology. I love it.
I love it too. I first encountered it when I was working at the Saint Justine Hospital, affiliated with the University of Montreal, and I was in Nicolas Deviker's lab. We were working on chemokine receptors, but we're heavily collaborating with Michel's lab.
Right.
And it was the time where I was shown how to do the column purification of cyclic AMP.
Oh, really?
In Michel's lab back in the day with the postdoc of his.
Right.
And then this biosensor came out for us to measure cyclic AMP. But before that, we were collaborating with them, measuring homo and heterodimerization of CCR4 with CCR2.
Right.
And I was following one of his postdocs who showed me the art of BRET. And I was hooked since then.
Yeah. It's a great technology. I love it. It's awesome.
So you use it. I saw that you had a paper come out with all of these G protein tools, biosensors,
right,
to use. And you usually look at G protein activation using BRET, or what are the other?
G alpha and arrestin.
Got it.
Yeah. So we're trying to, my sense is that so a lot of this is devoted to, ultimately creating biased ligands. And so we want to get as close to the transducer as we can without getting downstream. And so we want to get that initial event. The nice thing about both the arrestin BRET and the heterotrimeric BRET is that they capture basically that initial event. They're relatively insensitive to receptor reserve, which is amazing, So we think for analytical type studies, it's the way to go. So we've we still do lots of second messenger assays, but it's mainly most everything is basically BRET, or for screening we use Tango. So we developed this. We can screen all the GPCRs in the genome in a single 384-well plate using the Tango technology. So we use that a lot for screening, and of course Flipper, calcium for screening as well. So they're fast. You get a lot of false positives with them, but it's it's fine if you have a.
I was going to say the calcium assay is a tricky one too.
Yeah. It's fast. And the thing about screening though is you really want to assay that has a high signal to noise, which calcium does, and the Tango assays do really high signal to noise. And the penalty you pay in screening is not the false positives, it's the false negatives. So those are lost forever. So it's better to have an assay that picks up false positives, so you don't throw anything away, because we can always, a lot of our stuff is automated basically. So it's, a single postdoc typically would do 100 plates in a day, 100 384 well plates in a day. So just stack them up, the robot runs them. So running the assays is not a problem. We have, machines that load the buffers and all that stuff. So we just want to make sure we're not missing anything. So we'll accept a little bit of noise on the front end as long as we have, assays like BRET, which are very, very specific, very sensitive, to validate those actives down the road. So.
That's fantastic. You seem, well, you have a big lab, but you also seem to have a very organized and very compartmentalized set of tools that allow you to gather the information as soon as possible and also process it to get to where you want to go, which is understanding how these receptors actually work and possibly design drugs that would do one thing but not another.
Right. Yeah. And it seems to work.
I'm sorry?
It seems to work.
Which is great.
Yeah.
Great. One of my questions was, do you think GPCRs are still a good target? Obviously they are.
Yeah, they're great.
What do you think the challenges are at this point in order to develop better drugs targeting GPCRs and faster as well?
So, I actually get this question asked a lot by people in the biotech and pharma business, So yes, they're great targets. They're druggable.
Accessible.
The problem always is not in making the drugs against GPCRs. So what I tell them is, if you have a pathway, if you want a pathway specific drug, you can get it. It's not difficult. We've done it many times. If you have sufficient resources, primarily medicinal chemistry, the assays now are really good. You do need someone there that knows something about GPCRs on the team. That's very helpful to have somebody that actually knows the field. The problem, though, is deciding the indication, so the therapeutic indication, and the target. So the problems in drug discovery these days are not because you're not getting sufficient chemical matter. So we can do that. We can solve structures. So we're doing this now. We're solving structures. We're synthesizing new drugs. Then we're solving a structure with a new drug in like 3 weeks' time. And then we're making modifications of the drug and doing the iteration again by cryo. So it's resource intensive, but it's doable. The problem still comes down to, though, you have to choose the right indication, and that requires knowledge of biology that you probably don't have. Okay. So that's the problem. I think the reason that so many things fail is not because the chemists didn't do their work, but because the hypothesis was basically wrong. The idea was, if we make a drug against this target, it will be useful in this disease. And that just may be the wrong it typically is the wrong hypothesis. And I don't know of any useful ways of deciding how to connect the disease and the target, the disease and the therapeutic target. Okay, so this continues to be the problem. What I tell people is, and this is my my belief still, is that every GPCR is good for something, basically, right? If we make a drug against any GPCR, it's going to be good for some disorder.
Yeah.
We don't know what that is. That's the problem.
Oftentimes we don't understand the biology, the underlying biology of that pathology.
Right.
And also the the how much that GPCR contributes to that.
Right.
I was talking to someone earlier this week, and we were talking about depression, and actually it was more, it was migraine. And the problem is that most likely the brain of people who are suffering from migraine has a different biological landscape compared to someone who doesn't have migraine.
Yeah.
So targeting these GPCRs in the context of migraine is more difficult because you don't understand fully the biology of what's happening in that environment.
Right. And that's usually not what a drug company wants to hear.
Definitely not. I mean, I don't think anyone wants to hear that. Oh, you should, create a bias ligand for beta-arrestin and at this receptor, and all your problems will be solved.
All your problems will be solved, yes.
No side effects, most importantly.
Instead, I would tell them you need to spend 10 years finding out what the biology of this condition really is. Exactly. So they don't want to hear that. So that's one issue. The other issue is that for, complex diseases, which are the most common diseases, the ones that I'm interested in are neuropsychiatric diseases. Mm-hmm. So schizophrenia is a really good example. The genetic landscape is so complex that it's a drug that hits a single target is, never going to be, the best drug, basically. Yeah. You're going to have to make drugs that hit multiple targets. And they don't want to hear that either, basically. That's not what the chemists want to hear. But in fact, the drugs for schizophrenia in particular hit multiple targets. And for most, the ones that are most effective hit many targets, like 50. And clozapine being the most effective, at least 50 targets. And for other complex diseases, typically people are on multiple medications, basically, that are hitting multiple targets. And so the idea, if you could make a multi-targeted drug, that would be great. Then the question is, what mix of targets? Which ones do you want to hit? Which ones do you want to avoid? This sort of incomplete knowledge. I must say there has been some success in that. So I published this paper in Nature now about 5 years ago with this brilliant team from Dundee, Scotland. It was automated design of ligands to polypharmacologic properties. So a really beautiful paper. And they now have a biotech company, Exsentia, in Britain, and they're doing great. I mean, they basically industrialized, designing de novo drugs that hit defined groups of molecular targets, and that's working amazingly well. So I think that will be really important going forward for complex diseases. Of course, the problem always is what targets to hit, and we don't really know that yet to any great extent. So.
From patient to patient, would there be any variation? Would it work the same way, patient A versus patient B? That's also something that needs to be further studied.
Yes, that is a big problem.
Not all drugs work the same way.
Yes.
on this, even if the patients have the same diagnosis, you never know how their bodies would react to that.
Yeah, it's a big problem.
It is. But I guess
that's another problem. That's one I'm not tackling.
I guess that's another problem for another day.
Yeah.
I wanted to ask, so you had mentioned that before you became successful and you got all your grants, most of your grants approved, you went through a series of disappointments. What would be your advice for junior scientists who want to contribute to the field, but don't necessarily have, 10 Nature papers coming out of their postdoc?
So I never looked at it in the sense of contributing to the field.
Okay.
That was never that's never been my goal, actually, to contribute to the field.
Okay.
My goal, as I think was clear, I love being in the lab, basically. It's just so much fun. And I love looking at data, as my wife, who's listening to this, can you would agree with that, right, Judith?
Yes.
Yes. I love data.
It's an understatement.
I love looking at data.
It's an understatement. It's your passion.
It's my passion. So it's what I really love to do. And I'm just fortunate that, through luck more than anything else, things have worked out. The only advice I give people is, sort of trite, but it's all I have, is that find out what you love and then try to do that. Find the thing that you're really, really passionate about. Find whatever that is and do that. And don't pay so much attention to the outcome. It's the doing that's more important. When my lab was struggling for money, and it was difficult to see how I was going to keep things running, I was just really excited about every little bit of data. So I just love looking at data and the latest structure or whatever. They can't, people in my lab cannot send me too much data. So I'm just fortunate. I would say I'm fortunate that I found something I really, really like to do. And it's not work for me. It's just having fun. Every day is a vacation day in the lab for me. That's the way I look at it. So if I could give any advice to people, I would just say try to find what that is. The chances of any one person having an impact in science are so small that that has never been my equation at all. And if we've done things that are useful for other people, that make their lives better, either as scientists or ultimately the world's population, that's a great benefit, unanticipated benefit of what we do. I would say, so that's all I can say is, I wish you luck, and have fun in the lab.
Yeah, it is true. I mean, as long as you do what you like to do,
Yeah,
you end up not working a single day in your life.
right.
And if the goal is important, the way to get to the goal is also very important. It's an adventure. At least that's the way I see it.
Yeah, my goal has been to have fun in the lab.
And you've done it well, and you still seem to have
lots of fun.
Which is fantastic. We talked about the different projects that you've worked on and the discoveries that you've made. Were there any moments during that time that you discovered something, or you encountered something in the lab or within the scientific field that you can characterize as an aha moment? And if you could talk.
Yeah, so there basically have been two. So a lot of what we do is sort of discovery, historically has been discovery-based research. So non-hypothesis generated research. And we do that basically because I'm really bad at generating hypotheses. So my hypotheses usually are either wrong or they're trivial. But I would say the first sort of thing that was sort of mind-blowing to me was when so there's this plant hallucinogen, salvinorin A. I don't know if you know of this, salvia. So there's this hallucinogenic plant people smoke sometimes called salvia. And nobody knew how it worked. And so we discovered it was a kappa agonist.
Wow.
And when I discovered it, I had gotten the compound from this guy who was selling it on the internet. Okay.
Okay.
And I told him that it was the kappa receptor. And he said it cannot possibly be the kappa receptor because, as you know, kappa agonists cause dysphoria. Okay. And I said, well, I've been in the opiate receptor field and they're known to be hallucinogenic as well. And he said, no. He said, I'm going to disprove that it's the kappa receptor. Okay. So he told me that what he was going to do was to inject himself with naloxone and then smoke the stuff. Okay. And I told him not to do it.
It's not a good idea.
I said, this is a bad idea. Yes, you could die. And he did it anyway. And the next day he sent me an email saying, you're right, it's a kappa receptor. So,
oh goodness.
So I brought everybody in my lab to my computer, and I showed them the email, and I said, read this. I said, this is the only time in your life you'll discover something. You'll discover the molecular target of a drug on Monday, and then on Tuesday it will be validated in a human
trial.
Yeah. The human trial will be that night.
Well, I'm glad he survived.
He survived, yes. And of course, it's subsequently been validated as the kappa receptor. So that was that was a pretty amazing thing. The other thing was when we solved the first GPCR structure in the lab. That was amazing to me. So I had seen, with working with Ray Stevens, I'd worked on many structures up until that time.
Yeah.
And so I'd seen the structures and all that stuff, but to actually get it done in our lab was huge for me. So that was awesome, an awesome experience. And every receptor that we solve or that I'm part of now, they're all like really cool. Each one is amazing just to see, where this amino acid is and that one is.
You kind of close your eyes and at the end of the day know which amino acid is where.
Yeah. Each one is different, and they're all little beautiful snowflakes. So that's lots of fun for me.
Do you celebrate when you get a structure out? Is there any way you commemorate these?
No, we probably should. When we get a big paper that's accepted, we usually have a party. I throw a party for the postdoc, basically.
That's great.
But no, I probably should have, but we just never have done that. Yeah.
Well, maybe it's time for a new tradition.
Yeah. After COVID, we can have parties.
Yes. Yes. At the moment, it would be a Zoom beer hour or a Zoom party. You can have a bottle of champagne delivered to everyone.
Yeah, maybe I should do that. Yeah, it's a great idea. I probably should. I mean, for the postdocs, it's such a reward for them because getting these structures is like it's the most complicated and difficult thing in the world, is what I tell people. It's incredibly difficult to do, and particularly to get a good structure. And just getting the structure for them is like huge. So just to have, they don't need any. They would probably like to have a party, but it's
that might be, and I'm pretty sure it's not an additional incentive to get the structure out, but it would be a great way to close the chapter on that structure.
Yeah.
Fantastic. My last question for you, Bryan, today: if you have any job openings, do you usually post them somewhere? I need to mention that we do have a career page, Dr. GPCR. Career. So if you have any positions to advertise,
great idea.
The goal is to gather in one place all of us who love GPCRs, who work on GPCRs, so that people who have jobs openings can find the right candidate and not throw these ads out in the universe. And hopefully,
well, that's a great idea. So I should I would say jobs are potentially open all the time.
Okay.
In the lab. People should just contact me, even if I don't have a position now. So typically when people are applying, they don't need a job today.
Okay.
They're looking at, they're a student, they're looking to do a postdoc at some time in the future. So it's a big lab, things open up. people get jobs, basically. So I would just say, right now I'm not recruiting anybody, but who knows? Tomorrow, there are a couple people ready to be on the job market, and from my lab. And as soon as they get positions, there will be positions opening up, or in a year from now, something would open up. So I just say, just contact me. So great. So I didn't know about this, so I'll remember that. So can you send me
Yes.
email about that? That'd be
Yes,
great.
I can.
Yeah.
I can. The goal is to have as many job postings out there as possible. We're also scouring the internet industry jobs, which are very difficult to find if you want to continue working on GPCRs. But it's not impossible to find them. They're difficult, but not impossible.
There are some out there.
There are.
Yeah.
There are some out there. But yeah, that's the goal, is to have these as much as possible on one place, on one site, so that people can kind of shop for the best job that they're interested in. And this way, PIs and also companies can find the best candidates.
Yeah. So I know this company Accenture is looking for people.
So UK, right?
Okay. Yeah.
I've met at the last.
Oxford.
Yes, at the last GPCR retreat, I had met Kate, who was in your lab.
Yeah, Kate, yeah.
And she was presenting her poster, and she told me that she was in your lab, and she's at the company, and she seems to very much love it. Yeah.
They recruited my last 2 students. They recruited directly out of my lab.
Wow.
Yeah.
Handpicked them.
Handpicked them, yes. Stole them from my lab. Yeah.
Fantastic. I'll check out their website to gather the information for those positions, but I will also email you with a link and how to.
I know. So companies periodically contact me as well. So
okay.
I'll forward. I'm not going to say this information over the internet, but I'll forward it to you. Yeah.
We can cut this part.
Okay. All right. Okay.
All right. Thank you so much, Bryan, for being here.
Yep.
It was a great pleasure chatting.
Yeah.
We'll talk to you soon. Thank you.
Yeah. Bye-bye. Great service.
Thank you. Thank you for listening to this Dr. GPCR podcast episode. We hope you enjoyed it. Next time on the Dr. GPCR podcast, we chat with Dr. Arun Shukla from the Department of Biological Sciences and Bioengineering at the Indian Institute of Technology in Kanpur, India. If you'd like to sponsor us, please visit drgpcr.com/sponsors. We truly appreciate your support, which allows us to bring you podcast episodes, newsletters, and so much more. I'd like to thank Dr. Bryan Roth, Attila Forrest, Jin Chong, and Shivani Sachdev. Music by Rosa Bershish. I'm your host, Dr. Yamina Bershish. Thank you for the privilege of your time, and until next time, stay safe.